Welcome to the EW podcast. In today's episode, I speak with Alex Sonneborn. Alex is actually an old college friend of mine from Ohio University. Alex is currently a senior Ph.D. student at University of Texas Southwestern Medical Center. In today's episode, we take a dive into his dissertation, which I found extremely interesting. It's all about the effects that novelty and learning new things can have on the brain. The findings that Alex is coming across for his dissertation could have huge implications for treating addiction, depression, and even Alzheimer's. There's a lot of stuff in this podcast, but it all comes down to novelty and learning new stuff. You will have to excuse some connectivity issues at the beginning of this episode. For about the first 9 or 10 minutes of the interview, we were using a recording service that doesn't sound the best. We do make this switch to Skype around the 10-minute mark, and it sounds much better after that, so I hope you stick through. Other than that, this was a really great episode, and I hope you enjoy it as well. So without further ado, here is my conversation with Alex Sonneborn. Thanks for joining me.
Yeah, thanks for having me.
So do you want to just start off with who you are?
I'm Alex Sonneborn. I'm a senior Ph.D. student at the University of Texas Southwestern Medical Center in Dallas, and I study for my dissertation the effects of things like novelty and attention on learning and memory in the hippocampus and overall general well-being, I guess.
So what is the goal? Like, what is the ultimate goal? Why did you pick this?
So the ultimate goal to our research is there's an area of the brain called the locus coeruleus, and I know that sounds pretty sciencey, but really it actually just translates into the blue locus, the blue area, and because it looks blue under, if you dissect a human brain, there's actually kind of like a blue pigment to it. So this area of the brain was of pretty intense interest to a few really big labs over the last 3 or 4 years, in that normally this brain region was thought to release the neurotransmitter called norepinephrine, and this neurotransmitter is kind of like everywhere in the brain, but there was some hypotheses flying around that this brain region also releases dopamine, because this norepinephrine that is normally thought to be released from the locus coeruleus is actually created directly from dopamine. And obviously everyone knows what dopamine is. Dopamine is kind of our reward neurotransmitter. It's involved in a lot of different reward learning and social learning and things like that. So trying to figure out whether or not this dopamine is actually released from the locus coeruleus and whether or not this dopamine did anything under normal conditions was a huge target for a lot of labs, including Eric Kandel, who is a Nobel Prize winner, and he's kind of literally written the best textbook kind of right now available to any neuroscience undergrad course. So he's a pretty famous guy, and all these labs were kind of going after this question at the same time, and so we eventually kind of figured out that this dopamine is released from the locus coeruleus as a co-neurotransmitter with norepinephrine, and that was kind of a big deal since this locus coeruleus projects everywhere in the brain. I mean, there are synapses from this locus coeruleus at basically every brain region you can imagine.
So does that mean? Yeah, go ahead.
Oh, sorry. Yes, and this was like this is a pretty big finding that can, if you find the right way to target certain aspects of these neurons of the locus coeruleus, you might be able to really help a lot of people with certain things like depression, anxiety, and even so locus coeruleus often is one of the first brain regions to be degraded in things like Alzheimer's disease.
Interesting. So does that mean that whenever someone is like truly learning something, then that dopamine is being released?
Yes, I would say that. So our follow-up studies have shown a lot of follow-up studies. So we published a paper back in 2016, late 2016, that showed that this dopamine can be released. This locus coeruleus is a really, really important brain region for things like attention. I mean, this was the first thing. It was attentional processes, paying attention to something, was the first thing that this nucleus, the locus coeruleus, was actually studied for. And there's a ton of data back from way back when, the 70s and 80s and stuff, showing that this locus coeruleus fires when you're paying attention to something. This locus coeruleus is really, really active, and we showed in our paper that this when the locus coeruleus is the most active, that is when the dopamine is released, which is really cool. That dopamine tends to norepinephrine has been implicated a little bit more in kind of taking whatever brain circuits are active during attention. So what I mean by that is whatever you're paying attention to in the environment, norepinephrine helps to bring that signal, say something that you're watching or listening to, bring the signal above the noise of the rest of the brain. So it allows you to kind of really focus in on something. And once you can focus in on something and the locus coeruleus is active enough, then dopamine is released. And this locks into long-term memory whatever you were just paying attention to, as long as your brain thinks it was important enough.
So is the inverse of that situation, would that be like if someone is depressed or anxious, does that what kind of effect does that have on the locus coeruleus? Does it have a harder time of functioning? I'm just trying to imagine the connection between what it's doing and how understanding it and the neurotransmitters that it's involved with could help depression. Like what is that link like?
Yeah, so overall depression is still like a huge mystery, right? So we don't really know exactly what depression is, but it seems like a very brain-wide thing. And there are definitely drugs. So there are newer drugs called, so I think probably most people know what SSRIs are at this point, selective serotonin reuptake inhibitors. So there's a newish class of drugs called SNRIs. So this is selective norepinephrine and serotonin reuptake inhibitors. And these have been shown recently to probably, in a lot of cases, be slightly more effective than the like old school, I guess, SSRIs. I mean, obviously everybody's different, but these SNRIs would obviously they would target the locus coeruleus as well as all the serotonin neurons in your brain, and these have been really effective. So it turns out that the locus coeruleus, one of the things, so in our paper, the main thing that the locus coeruleus not only is involved in attention, so anything that is your brain kind of decides is interesting, or the sciencey term is salient. Salience is kind of like the science term that we use for interesting. So the locus coeruleus is really active for any sort of salient event. And actually it's been shown that mice, or if you do some experiments on mice, giving them novelty, new events are very, very salient to humans, of course, and even mice. So mice will actually, if you give mice enough new things to do instead of just having them in their boring cages all day, they'll actually show way lower signs of depression, way lower signs of anxiety and kind of social awkwardness, I guess. You can kind of say that in a mouse. So the locus coeruleus is really, really active during novelty. And there's like a huge, after our research came out and all the other labs that were working on that also published very similar findings. So novelty has been now really kind of a big focus of a lot of labs, and that novelty can actually be an antidepressant, like a really good way. So a lot of times, when a lot of people, they're working the same job every day, you go to your desk job, it's horrible. It's the same exact, you get in your habit, your same thing every day. You eat the same breakfast every day sometimes almost. It's really easy for humans to fall into these habits, right? That, the brain likes to be able to predict exactly what's coming next. But kind of maybe counterintuitively, I mean, we've known for a long time that the brain likes to predict, likes your environment to be really predictable, which is why you get into really, really mundane routines and stuff. But it seems like novelty can really, really activate the locus coeruleus and might be able to combat a lot of depression that people see just from their everyday jobs and 9 to 5 and years and years of the same thing every day.
It fires, or it becomes really active, this locus coeruleus, in response to very salient events or events that a mammal might find are really important to something, to getting a reward, to surviving something. And so this activity increase in response to salient events allows this means that this locus coeruleus is going to be very, very active in response to novel events. So novelty is something that's really, really salient to mice or monkeys or humans or whatever animal that even has a locus coeruleus, which is all mammals. It's very, very, very old brain region, kind of evolutionarily speaking.
Do reptiles have it?
I think probably, yeah. I would say that. I actually don't know. I only work on mice, but that's a good question. So novelty is really, really important. If you go back, if we're just talking about humans specifically, novelty becomes really important, and it should be something that humans, especially if you're a really, really ancient human, 10,000, 20,000 years ago, you're an ancient human kind of trying to figure out your environment, trying to learn more things about what's going on. Your tribe doesn't know, all that much about the surrounding area. You have to move. So this novelty detection from the locus coeruleus, like this increase in activity, is actually, it should produce some sort of reward, which should allow you to seek out this novelty.
It's like a natural selection kind of mechanism. That's like, oh, we're gonna the brain is just rewarding people who push the species further, almost, or like I don't know, find ways to survive.
It's a very human thing. I mean, mice kind of have almost the same response from the locus coeruleus, because it's, like I said, it's really old evolutionarily, so it almost works the same in every species. But if, like, really importantly for humans, I mean, the pursuit of new knowledge, I guess, would be like a really, really important function. And like the locus coeruleus is something that maybe not everyone can kind of gain the same benefit to the same extent, but some people, the pursuit of new information, the pursuit of new knowledge, that's like, sometimes you don't think about it, but that's rewarding. It's very rewarding. New things, just learning as much about your environment as you possibly can, is really something that humans need to be able to do. And so there's a natural drive to go do this. And it's in part mediated by the locus coeruleus. So the main dopamine nucleus, the brain region that produces dopamine and releases dopamine all over the brain, that is also really active for novelty too. So there are two. So paying attention. So the locus coeruleus kind of allows you to pay attention to this new novelty, decide where you want to go, what you want to go explore, and you get rewarded by partially a little bit probably the dopamine from the locus coeruleus itself, and then you get a lot of reward from even your dopaminergic circuits in response to novelty. And that's really cool. Normally people think of dopamine basically in response to drugs, things that make food, sex, drugs.
Right.
But it has functions that are a lot more important just for your basic survival, which is really interesting.
It's encouraging survival techniques, basically, in your brain.
Yeah, kind of that encouraging. Yeah, the best way, encouraging. Yeah, like you said before, pushing forward and kind of discovering new things, which is very philosophical. I mean, it's hard to prove, especially in mice.
Yeah, I totally get it, but the connection seems to be there.
Yeah, there's something there. There's something really cool there that, some philosophical questions are extremely hard to answer with.
I love that. I love whenever philosophy kind of turns to science, and you know, you have that intersection of like things that, you know, way back in ancient cultures or whatever, they would never think could be solved, you know, and we're getting close to solving some of these questions, which is crazy.
Yeah,
Um,
really, really cool.
so in the mice that weren't participating in novelty, that you said exhibited signs of depression, were you guys looking at their locus aurelius and seeing if that like degenerates or anything? Like if it's not stimulated, does it kind of deteriorate, or does it just lay dormant waiting for stimulation?
Yeah, so the locus coeruleus is kind of always on a little bit. It just there are definitely cases where um, deterioration leads to. I would say deterioration probably leads to more obvious memory deficits and attention deficits than actual depressive like episodes and stuff like that. But so we didn't in our study we didn't necessarily test the depression. I was kind of referring to other previous
Oh, okay.
studies way back in back in the literature when people were first doing this kind of stuff, looking at how the locus coeruleus and dopaminergic uh, other non-locus coeruleus dopamine circuits are involved in depression, and there seems to be a very very clear link between novelty seeking and depression, whereas sometimes like there are I've seen examples of mice that actually seek out novelty more fiercely, like more readily seek out novelty, are actually kind of more prone to being depressed. So they're maybe they're almost using novelty, it seems, in those under those conditions as a drug almost, and that's really interesting too. Like they have their whatever genetics these mice have that make them slightly more prone to depression also makes them slightly more uh, kind of needy for that novelty, that new novel experiences, and that's really cool. So there's some weird
Hmm.
connection between novelty and depression, and this isn't quite
And
teased apart yet.
and even addiction maybe in some ways.
Absolutely. Yeah, so I mean a lot of people would say that. So interestingly, and this is kind of totally off topic from everything else, maybe a little bit related, but so the locus coeruleus, so heroin, opiate drugs, heroin and morphine and oxycodone and drugs like that, they bind to opioid receptors in the brain. So your brain has its own opioid system, and these drugs will activate this opioid system, and it's an extremely addictive system, right? I mean, you can really get addicted to heroin and morphine.
Hmm.
Interestingly, the locus coeruleus actually has one of the highest, if not the highest, densities of opioid receptor uh, of the opioid receptors in the entire brain. And a
Whoa.
lot of people think that one of the reasons it's so addictive is because every time, uh, whenever these you take these drugs, it's doing whenever you take heroin or morphine, it's doing something to novelty circuits, and it's making so opiates tend to be inhibitory. So while this drug is there, while this drug is in your system, it actually inhibits these locus coeruleus neurons. And but for something like withdrawal, uh, the brain loves to be at a homeostasis. It'll upregulate, its firing rate. So whenever you take away
Hmm.
heroin and morphine, now you have a really, really overactive locus coeruleus system, and it might end up like there's something that it's doing. It's making you focus really hard uh, on heroin and morphine potentially. As after these circuits have been activated after you take the drug, so a lot of brain circuits are activated when you take a drug, and addiction
Right.
kind of starts after you take that drug. You get a big boost of either norepinephrine or dopamine or sometimes even opiates or cannabinoids, and um, this really really can change your brain chemistry and make you fixate on that drug, as those circuits of drug taking are locked in with these big
Hmm.
with these like memory generating circuits like the locus coeruleus and the dopamine circuits such as the ventral tegmental area.
Okay, so let's just I just want to make sure I have my head completely around how the locus coeruleus is working.
Sure. I mean,
So
you and me both.
yeah,
I'm not 100%
okay.
sure,
But the basic understanding of it is a person is in need of, we'll use a mouse since that's what you're studying. A mouse is in need of novelty, and that is activating the locus coeruleus. It's making it active, the need for novelty.
I guess not necessarily the need, but the when you notice something is new, I guess.
Okay.
Once you come across something that is new, the locus coeruleus will really increase its activity.
Okay, so it's once that new thing is encountered. So whenever someone is addicted to something and they don't have it, their locus coeruleus would be not activated, or it would be.
Yeah, so that's pretty weird. So it would be activated. So well, depending on the drug. I mean, for opiates, so every drug works a little bit differently, but for opiates, that's how it would work. And no
Is
one
that
really.
because oh, sorry, no one knows.
Yeah, I mean,
Okay.
it's because there's such a high density of opiate receptors in
Okay.
the locus coeruleus, and opiate receptors. I mean, this gets into some kind of convoluted stuff that's really hard to explain over a phone conversation, but
Yeah, for sure.
but yeah, so opiates are typically inhibitory, and whenever they're there, they're going to over inhibit the locus coeruleus. So whenever you take them away, the locus coeruleus has kind of upregulated its activity in order to combat. So that it likes to be at like a happy homeostasis. You know, everything in the brain kind of does this, and this is what drugs do, right? They take away this homeostasis. Your brain tries to make a new homeostasis. So when you take the drugs away, whatever mechanism it used to create that new homeostasis is still active for weeks, and this is how withdrawal happens.
Gotcha. Then okay, I think I got it a little better that time around.
and it's really confusing as to what the locus coeruleus is actually doing. So the locus coeruleus also sends axons or sends connections over to the VTA, the ventral tegmental area, which is the dopamine nucleus. So it's probable that increased activity in the locus coeruleus could cause more dopamine to be released, and the dopamine in this other brain region, the VTA, is really, really important for reward-seeking behavior. Once this dopamine becomes active, once this ventral tegmental area becomes really active, the mouse will you almost, I mean, and this, you see this all the time, even in humans. I mean, this is a kind of a subconscious thing, right? This dopamine is overactive, and you might seek out something, and in this case, you would seek out heroin like very readily without even really thinking about it. And it's just this overactive dopamine system, overactive locus coeruleus system, overactive dopamine is probably overactivating your dopamine system, which would lead to some sort of seeking behavior. So you would seek out a drug in order to just subconscious. I mean, you're not even thinking about it really.
That's
you might
crazy.
not even like taking it. You might think like, I don't even want this. I don't know why I crave it so badly, but you do.
Right. I think having an understanding of like the stuff we're talking about, and understanding where cravings come from and what they actually are, can help, combat them. I'm not saying it's a cure-all for someone who's going through heroin withdrawals, but maybe, someone who loves chocolate too much, understanding that they're just responding to, brain chemistry is kind of an interesting thing.
Absolutely. And I think so. I actually teach right now a psychopharmacology class at Texas Christian University, TCU. And the one thing I try to really get the undergrads to understand is like, a lot of them are coming from biology backgrounds or psychology backgrounds where really all they're doing is memorizing stuff all day and like memorizing facts about biology. And I really try to get them to understand, like, look, this brain chemistry is like, you can use this in real life. Like this is something that, it's not just a fact I want you to memorize. This is like real life. This is happening all the time. There's a huge opioid crisis right now. I mean, we don't just learn about opioids in that class, but this is like something that's really, really important. And obviously, back to your first point, there is this, there's something that's pushing you to seek out things that maybe are bad for you, especially drugs and alcohol. But the cool thing about like mice, I don't know, mice probably are screwed, once you get a mouse addicted to something, because they don't have kind of the cortex, they don't have the human cortex that can
Right.
say, look, I shouldn't be doing this. Like,
That's
this
interesting.
is my, I can stop. Like, a human can say, it's really difficult, though. I'm not saying anyone who's addicted should go just be like, oh, I should just stop and do that, because that's not really the case. But humans can realize what's going on. So humans have this cortex, and this gets into really, really philosophical stuff about free will and all that nobody knows anything about. But
Oh, I love thinking about that, dude. I've been checking out so much of Sam Harris's writing and talks
yeah,
about
yeah.
free will, man, and it's just mind blowing. And I think it all comes back to this: that just chemicals firing in your brain, making you act a certain way. Not to say that you can just blame every action on your brain, but I mean, to some degree, yes, you kind of can. You kind of can say this is just my brain.
Absolutely. And it seems like with our so the your reward systems, or your reward center. So the dopamine systems in your brain, if you're going to get addicted to something, usually the dopamine systems will increase their firing in an area of the brain called the nucleus accumbens. And this nucleus is kind of the brain region that's really doing the deciding, quote unquote, of like, is this something that I'm going to seek out very heavily in the future because I was rewarded by this drug. So you really can trick your nucleus accumbens into seeking out a drug that elevated dopamine in the nucleus accumbens, and it's this really bad positive feedback loop that kind of spirals out of control. You keep taking the drug, that's more dopamine in the nucleus accumbens. You keep rewarding yourself for seeking out this drug, and it's really bad. But the cool thing about the nucleus accumbens is it receives a really, really dense projection, really, really dense synapses from your prefrontal cortex, and a lot of your cortex, your cortical areas that are just kind of generally thought to be involved in decision making, problem solving, planning. So you can, that's good news for humans, because we can say, I don't want to seek. Sometimes you get an urge, and you can suppress that urge, right? Everyone's had this experience hundreds of times in their lives, and that's probably your cortex saying, no, don't act on that urge. It's like your cortex and your more primitive kind of subconscious are fighting over whether or not to do some sort of behavior, and that's really interesting. I mean, that's like, that's philosophy at work in biology.
Yeah, that's super interesting. So what kind of experiments are you guys running now to test this? Like, I'm assuming you're still using mice.
Yeah, we still use mice, and that's a really good question. So we figured out that the locus coeruleus could also release dopamine, and that the kind of logical and a little bit incremental next step there is how is this dopamine actually being released here? And it turns out that it's not normally, neurotransmitters are released with vesicles, through synaptic vesicles, and it's hard to, you'd have to take a whole class getting into this process here. But my point, I guess, is that we wanted to go after the mechanism under which this dopamine is actually released, and there's some good evidence here that it's through some weird, very strange kind of non-canonical neurotransmitter release mechanism, where it's actually reversing what's called the norepinephrine transporter, which is what's normally it acts as kind of like a vacuum after neurotransmitters are released, after norepinephrine is released, so that it recycles it. It's like an energy conserving. You don't have to keep making new neuron. You can actually just reuse and recycle the ones that you already used, which is really cool. It's a whole nother story, but
Is that true for all neurotransmitters, or just
yeah,
norepinephrine?
most neurotransmitters will have some system to recycle their neurons, because the brain already is using so much energy that it's any kind of step up it can get to really conserve as much energy as it can, it'll take advantage of.
Evolution, baby.
Yeah.
So cool.
Yeah, it's really interesting. Yeah, and I mean, plenty of drugs work on, obviously, serotonin, SSRIs. These vacuums are what the SSRIs are blocking, so that the serotonin can't go back up into the serotonin neurons. It sticks around in the brain for a little bit longer.
Interesting.
Yeah. So
So
we
go
found
ahead.
that these vacuums actually might be kind of reversing their vacuum activity and actually spitting out a
Hmm.
neurotransmitter. And that's what we're working on right now. So the whole point of trying to figure out the mechanism of this dopamine release is, since this locus ceruleus is involved in so many affective types of behaviors and reward types of behaviors and even Alzheimer's, it would be really useful if we could know some exact target to kind of really single out with a drug. And this is a lot harder said, or a lot easier said than done. Sorry. And it would take probably decades to actually complete. But you could really kind of go in and really design a drug or try to figure out some drug once you know the actual mechanism of release that could help a lot of people with disorders that are very, very common these days. Alzheimer's is increasing. Depression rates, especially in millennials, are skyrocketing. I mean, it's really bad.
Yeah, it is tragic.
Yeah.
Is the finding that the vacuums spitting out norepinephrine, right?
Mm-hmm.
Or
Yeah,
dopamine.
the
Sorry.
transport, yeah, they're called transporters.
Transporters.
Is that a very rare thing, I'm guessing, to see that the reverse action happening, or is that something that happens with other transmitters as well?
Yeah, it's a really good question. So this idea didn't come out of the blue for our lab. There was another lab that found that the dopamine transporter on the dopamine neurons in the ventral tegmental area can actually reverse transport the dopamine as well. So we think that the dopamine could be just coming out of the locus ceruleus in the same way. So that's kind of that's where we made that. There was a definite logical step there. It wasn't like we just randomly tried to do this, and that's a really good question. I mean,
what would be the next step? I mean, so ultimately the goal, the end game, is to find some sort of drug or application in the real world for real people, to fight real diseases. But what are some of the steps, I guess, like that are still in the way between where you guys are now and where you are going?
Yeah, so really importantly, we would have to figure out this mechanism first. So once we have that mechanism, we can try to see what types of drugs. I mean, there are definitely drugs that already exist that we could maybe use to just very crudely test certain behaviors. So there are behaviors you can do, and this is pretty sad, and obviously mice are kind of the first line of, is this going to work, right? And you obviously kind of are subjecting them to some serious amounts of stress. Anyway, but there are a bunch of behaviors you could do while manipulating this mechanism that we find. You manipulate it in a bunch of different ways, and you can try to figure out what way most effectively kind of would eliminate or alleviate depressive-like symptoms in mice, because you can get mice depressed pretty easily. There's a bunch of different paradigms. You can use another mouse. It's called social defeat. It's a really interesting test where you basically take like an alpha male and you have this other mouse that this alpha male just bullies. It's very strikingly weirdly similar to some human interactions, but there are tests where you make a mouse struggle and you measure how long it takes for that mouse to stop struggling. And if they stop struggling really quickly, that means they've kind of given up, right? And so the quicker they stop struggling, the more depressed they are. I know that's pretty sad, but a lot of times it's come up with some really interesting findings that have made it into the clinic. And yeah, it's a useful thing, but you could use things like that. And obviously, for diseases like Alzheimer's, what I personally am slightly more interested in is how this dopamine that is coming out of the locus ceruleus in this weird way is actually able to affect memory, and you might be able to even enhance certain types of memory, and especially like novelty dependent memory, like in Alzheimer's, right? People can't remember new things. They can remember things that are that they've known for their entire lives. I mean, until it gets bad
Right.
enough. But at least initially, these people can't remember anything new. So
Mhm.
there might be some way to manipulate. And like I said before, the locus coeruleus is one of the first brain regions to degrade in oftentimes in Alzheimer's, and this could
Hmm.
be, uh, activating some or allowing the locus coeruleus to kind of become effective again at remembering novel things could be like a really important way to help a lot of people with dementia.
Would continuing, like throughout one's life, just continuing to subject oneself to novel things, do you think that would have an effect on keeping away Alzheimer's? Like, or is it not really dependent on how a person lives their life? Does that make sense?
Yeah, that's a really, really good question, and it seems like that's at least part of the equation. I mean, obviously, if there's some weird genetic
Right.
defect that you unfortunately have, there's not a lot you can do. And some people get Alzheimer's when they're 50, but yeah, I think that this has not been studied all that much. I mean, it seems like there are definitely correlations between, keeping mentally active, at the very
Hmm.
least, keeping mentally active. I mean, that's not necessarily novelty. Um, but I think it would be a really interesting study, really interesting to do a longitudinal study where you almost, and this would cost a ton of money, but you basically take humans and you just keep sending them out to experience new things and get new degrees and stuff like that, and then you just take some control group. I mean, this is like not even a possible experiment almost,
I
because
mean, it could be. You could find people that are already living their life like that, and just study them. You
Yeah.
know.
yeah, take that all the way out until these people are 70. Maybe they're in their mid 20s, late 20s, and all the way out to when they're 70 or something, and take the vagabonds versus all of the you know the people in 9 to 5 desk jobs, and
Yeah.
that would be pretty interesting to see the rates of Alzheimer's under those conditions, and that would take a lot of work from what I mean. That's a huge longitudinal study over. It would have to be I mean, 50, 40 or 50 years almost.
How many people would you need in the study like that for it to be legit?
I mean, I would say at least. I mean, you can calculate it. There are definitely ways to calculate It's called a power analysis, but uh, at least a few hundred in each group, and that would be really pushing it. I mean,
Wow.
it would be probably more of like the thousand into the thousands. You'd need like 1,000 people.
Yeah, that might be a little difficult.
It would be tough, yeah. It would be really hard. So mice are cool because they're all the same. They all have, well, they should all have the same exact genetic makeup because you basically inbreed them until they're all exactly the same. So it's really easy to control for all these things for mice, but the variability would be enormous in humans. I mean, it would be kind of off the charts, which is why you need so many. Uh, but yeah, I think that type of study would need to be done. That definitely has not been done in humans, to my knowledge. I mean, maybe someone has done it, but I I,
This.
I, I don't look at the clinical side as much.
I recently talked with this guy, Michael Lavery, and he's got this program called Whole Brain Power. I actually wrote a little follow-up blog post about it today, just kind of detailing my experience with some of the training methods in it. And one of his things is having people engage the brain through their hands. So he has a bunch of practices that are left and right hand, like left hand mirror writing, but all these different just drills basically that require you to engage your hands on an ambidextrous level, both hands. And he claims, and I don't really know the science on this, maybe you can shed some light on this, but that doing these drills and doing stuff like this can help fight Alzheimer's. And it seems like, based off what we're saying, there might be some like valid science behind that.
Yeah, I mean, it seems like that would be something that's almost like reverting your brain back to like how difficult it was to first learn how to write, like that's reverting your brain almost back to
Right. And that's
just
what he
that
says is like you're having
juvenile.
Yeah, he says if you're right-handed, your left hemisphere is basically teaching your right hemisphere how to do something.
Yeah.
I
Yeah,
don't know.
that's really interesting, and I don't I like, again, unfortunately, I really rarely read anything about kind of like human studies or clinical stuff. But I would be willing to bet that there's probably some good evidence that things like that, you'd have to really, really, really try to find some like full brain engaging experiments like he's talking about. But definitely anything's possible. That seems like something that's not like probably not a scam or like anything like that. One of the theories of why the locus coeruleus is kind of the first thing to go during Alzheimer's disease and other dementias is because maybe people use it too much. So yeah, there's obviously all, there's until you can find some causal relationship between something, which might not even be possible for Alzheimer's. It's really hard to say exactly what's happening. Like for something like the locus coeruleus, it's involved in so many different things that you just do day to day. I mean, you use it a lot.
Is it part of the hippocampus?
Locus coeruleus is part of what's called the pons. So it's part of your brain stem. So yeah, this is again like your brain stem is like really deep. I mean, these are every mammal has a brain stem. This is. So this is like really old, and it projects really densely to your hippocampus. Like it sends a lot of synapses to your hippocampus, and hippocampus is definitely so. That's what my work is done in the hippocampus, because there's so much locus coeruleus axon terminals there. But yeah, I don't know. I would say that it's just, locus coeruleus is really, if you're really stressed out all the time, you probably have an overactive locus coeruleus. Like it's firing too much, way too much, and it's hard to focus on things. And if you've been really stressed, right, it's like you can't sit down and read if you're super stressed out. You're just the locus coeruleus isn't functioning properly. You can't focus on one thing. It's like switching between all kinds of different things at the same time, and so just overactivity. If you have too much stress in your life for 30 or 40 years, that might be a problem for your locus coeruleus, which is why it degrades so quickly.
If you guys were to figure out say you reach the end goal of finding like a drug that interacts with the locus coeruleus in a way that you've been looking for, is the brain chemistry or brain anatomy between a mouse and a human close enough that you could test that on a mouse and suspect there are similar results with a human? Are they similar enough?
I think the locus coeruleus would be similar enough. I mean, it depends on what you're trying to claim that it's doing. So it would be, you could do all of the cognitive testing you want on mice, and it's just not going to be the same, right? The human cortex just crushes the mouse cortex in any sort of learning task or something like any complex behavior. But yeah, I think those primitive brain structures like that are probably not so different across many species, like really any species. And I think you could at least make some generalizations. Yeah.
That's so interesting, man. It's crazy the kinds of questions that, like we were saying earlier, are being answered just by looking at the brain.
Yeah, philosophical type stuff that we're now learning biology about. Learning about the biology for it. It's really interesting. I mean, and it's only going to get more and more kind of abstract. Like the question, or like the data is really hard, going to become really hard to analyze because it's so weird and abstract. You're already really seeing a lot of that is in neuroscience these days, where there's so much data that it's really hard to just, like, the brain is so noisy and there's so many things going on at the same time, like in parallel, that it's hard to really tease it apart.
What kind of things do you take? You mentioned earlier talking to the students you teach at TCU and trying to get them to see how applicable some of the things they're learning are in your class. Like, what kind of things do you take away? Like, what are some examples of things that you're just like, oh, how are you not seeing this? Or,
Yeah, I think one big one, like the one that we've been saying that we talked about earlier, is that your brain, being aware that there are a lot of things that you don't really have control over, that there's a lot that just goes on under the radar that you could have control over if you really think about it. And this, this is totally aside, but that's kind of one of the benefits of meditation and stuff like that is learning how to take just things that occur in your body and think about them and kind of just uh, either let them be, or you can learn to really manipulate them and have a lot of willpower, quote unquote. Some people might argue that you don't even have any free will, but just understanding that there are all these processes that really you can have control over if you try really hard and think about it, but it takes practice. And sometimes, if you obviously take the wrong drug, it becomes really, really difficult once you throw off these kind of under the radar systems. It becomes really, really difficult. The stronger that these systems get, you're really, what you're doing when you're taking a drug is you're making these associations. Your brain is just an association machine, right? It's making these associations, and you're making these associations so unnaturally strong with a drug that it becomes really, really hard then to go in there and say, okay, I don't want to do this anymore. I want to suppress this urge. And just knowing that I will never take drugs like that, because you just know that you can crank up these systems so high that you end up losing control over them.
Mm-hmm. Do you meditate regularly?
Not regularly, not anymore. I've been a little too scatterbrained recently trying to finish up school here, but I used to, and it really, I need to get back into it at some point, because I think it was extremely useful back when I was doing it. But
is that a big thing, like in the lab? Do people that you work with, are they big meditators? Is that a big thing?
I would say probably not. I would say that you could definitely find one or two people, but overall, it seems like graduate students are the postdocs, after you're basically just still a student after you get your Ph.D., and you keep doing pretty much what you're doing, except you're expected to produce more stuff. So these, it takes a strange breed of human almost to do these types of jobs, and it's really high stress, and I don't know. It seems like these people are a little bit more kind of stressed out than meditatively calm.
Yeah, and I guess you could speak to that right now with your state. Yeah.
I almost might not even be able to meditate if I even tried at this point. There's so much going on. But
that's your tempus aurelius. Did I say locus aurelius? Tempus locus aurelius, just too active, right?
Yeah, that for sure, probably. I mean, knowing that is really kind of unnerving to me, and I'm like, I've been so stressed for a few years, and I'm like, well, am I gonna get Alzheimer's if I stay this stressed my whole life? Like, I can't. Am I gonna get Alzheimer's? Is my locus aurelius just gonna degrade by the time I'm 60 or something like that? Because there's all this stuff going on, and I hope. I mean, it's not just locus aurelius too, right? So your entire fight or flight stress response, like in your entire body, ends up kind of being activated. But that's a whole other story.
The sympathetic nervous system.
Yeah,
right?
Sympathetic nervous system. Absolutely. The one thing to take away from my research, anyway, the one thing I hope people could maybe at least use is the novelty thing. I think that it's so easy. So just like I said before, your brain loves, and this is kind of why people love music so much, right? It's a predictable pattern. Your brain loves patterns. Your brain loves predictability. But that's kind of, I guess, since your brain evolved to love being able to predict stuff, there needed to be some extra reward, right? To go out and seek novel things. So you can break out of this kind of cycle of just wanting this, or just trending towards predictability. You can break out of it, and if you're aware of it, you say, oh yeah, I guess I do the same stuff every day, right? I mean, this probably is a thing that a lot of people in the country, definitely in America, are doing. Just kind of subconsciously going through the motions.
I can speak to that sometimes,
Yeah, me
just
too.
getting caught in a rut. And I work from home, so a lot of days for me is waking up from the bed and walking to my computer.
Yeah,
And
you
then,
maybe don't even go outside. Yeah,
yeah, sometimes I don't. I've been trying to make better habits of that, but sometimes you just get caught up in it, and then next thing you know it's the end of the week and you haven't left the apartment.
yep. Yeah, I completely agree. I mean, I just think being aware of that and being aware that novelty is actually rewarding, and it can kind of probably be rewarding enough to really kind of give someone the spark that they might need to get out of a rut, get out of something like that. Just try to make it every day. I mean, you can't just go do one thing a week. It's probably would have to be almost every day, and it could be something as simple as like you go for a run outside and you run a different direction this time. Like if you just break out of that rut. And I know that sounds really obvious, but there's now plenty of data that kind of support that this novelty breaking out of that rut can actually be, jumpstart your brain almost. I know that's not a very sciencey thing to say, but I really think that there's something very therapeutic about this novelty and that can be used, and a lot more has to be learned about it, of course. But just like anything, brain science is a pretty young field, but I really think there's something useful there, and I think that humans evolved to kind of want that novelty, or
Subconsciously.
yeah,
Yeah.
subconsciously kind of. I mean, I know that seems almost exactly the opposite of me saying humans evolved to enjoy patterns and want to get in a routine and be a pattern, but the patterns don't provide any sort of um, like reward, right? So these patterns that you get in, and it's almost like anti-reward. It seems like eventually you just get into them. There's no activation of any sort of reward pathways over time. I mean, sometimes people, and I've experienced this too, where you start out, a job is really interesting, and eventually maybe you like your paycheck, and eventually your paycheck just isn't rewarding anymore. You're just like, I'm just doing the same thing every day. Like, sure, I get money, but that reward is just gone. I mean, there's nothing rewarding there. So there's a system in place to kind of take that. It's almost like your brain understood itself a little bit and understood that there was some sort of like your brain knows that it likes to get into these ruts and get into this just very familiar territory, just stay familiar, stay in this familiar territory. But then it kind of, something happened and it said, oh, actually, let's make novelty rewarding. So people learn new stuff. People go explore new things. They figure out, where the best escape route is. They figure out what the best animals to eat are in the environment, what the best berries to eat are, what trees can I,
how to make
plants,
fire.
Yeah, like what plant, yeah, how to, exactly, like what happens if I bang these sticks together in a new way that I haven't done before. Like this. So your brain figured out some way to get out of that loop of just sitting around all day. And I know that's not a very sciencey way to put it, but I really think that that's what happened, and it's really interesting.
I totally see where you're coming from, and it's really cool that you're working on science to kind of progress that area. That's awesome.
Yeah, and we'll see what happens. I mean, hopefully in my lifetime I will see something. I can't imagine that novelty just not only is it, I mean, there are entire economies based on kind of novelty, right? You go to a different place, tourism is novelty, like this is something that we've monetized. It's like this is getting really political, and maybe I shouldn't even say this on a neutral podcast like this, but it's almost like, we did realize that novelty was really cool at some point, but then democracy just, greed just monetized it. And it's like, oh, well, we're just going to make it, cost a bunch of money and stuff. And like people obviously will pay for it because it's rewarding. Going to a new place is rewarding. It's almost priceless. It's in some cases.
For sure.
And it's just one of those, like, novelty. You never think about it. It's, again, in locus coeruleus, it's a really ancient structure. It's subconscious almost, but this reward is something that, is very, very obvious to see. People paid so much money to go do novel things. And, this is akin to a drug almost.
It's kind of makes me think of like the value in having a hobby. You know, if you're stuck at a job, having something that does provide novelty and a challenge to you,
Yeah.
can be, sounds like really beneficial to your brain, even
Yeah.
just to yourself as a well-rounded human, you know?
Yeah, absolutely. Definitely helps with the well-roundedness and I mean, again, yeah, you can go. I mean, that's what everyone. The first thing everyone you always see like on blogs on the internet and stuff like, how do I'm stuck in this rut. How do I get off? And they're like, oh, people, any just random internet strangers go, oh, you should go get a hobby. And it's like, well, that's probably. I mean, that's a good. That's a good first step. I mean, if you're.
Right.